Hi, Professor. My name is Nidia Pozo. This is the week 2, and in this particular experiment, we're doing the bacterial enumeration dilution. So the goal of this is that we will begin to dilute the bacteria grow in the tubes. So we have 6 tubes that has mark from 10.1 to 10.2 to 10.3 all the way to the 6, and we also have our agar plates, which basically are labeled with the same numbers, 10, 1, 10, 2, 3, all the way to the 6. So the idea of this is to show how from a single cell's growth you can in half hour have 30, and in a couple of hours or 10 hours you can have 1,000,000 of these bacteria. So when we dilute it and make it thinner and thinner, the goal is to try to ending with the correct count of 300 of the growth of the bacteria, how one single cell can divide into 2, creating colonies, and only in 30 minutes. We, of course, have cleaned our surface, have prepared the gist, have prepared the plates 1 hour before the experiment. We have distilled water already in the tube, 2.25, to begin the experiment. And we have our pipette, our water, so everything is ready to go. So the first thing is we have our gist, and then it's required to take just the 25, which is as the first line of the pipette, and then we're gonna take the tube 10.1. So this is the 10.1 that it has already our distilled water, and we're gonna put the 25 for the gist. Now the idea to do this is to wash very well all the gist from the 2.1, and then from the 1 we're gonna dilute to make it thinner and thinner as we go to the other tube. So same process, we're gonna take the 0.25, and then we're gonna take tube number 2, and we're gonna do number 2. So I'm gonna put 10.1 back, and then from number 2, same process, to take to the first line, to the 25, to number 3, and we put in the number 2 back, and we're gonna go ahead, and the continuation keeps going until we can get clear from the enumeration of bacteria. So we're gonna continue all the way to the 6 tubes. So go ahead and pause, cameraman, so I can do the shorter version. So we have done all the transfer, diluting from one tube to another tube to another tube to make it thinner and thinner to ending in the 6. So now our pipette has to be really clean, and the idea is to transfer to the plates. An individual plate will have the numbers. So from the 10.1 to the 6, the one it's going to be here, and the same process. We want to just tilt a little bit to not contaminate. It says that we only need 4 drops. So this is the number 1. One, 2, 3, 4. Then we're gonna wash it because we want to be sure that we don't transfer the dilution to the plate number 2. So that's wash. Then we're gonna go number 2. Same process, I only need 4 drops. And we're gonna go to number 3. And we go into number 4. Four drops. Number 5. Same process. You want to be sure. This is number 6 of the dilution. Tilt it a little bit, 2, 3, 4, and of course this has to be done. So now we just want to take and move the liquid inside of our plates. Be sure that it's expand completely. It has to be in incubation for 48 hours. 12 hours, and then turn it to allow the microbes to grow properly and evenly. So as of right now, I'm going to check this back in 12 hours and then turn the plate to allow the bacteria to grow properly. The idea to move it is to evenly put it in the agar area. So this conclude for now the bacterial enumeration and dilution. And we'll come back after 12 hours, review how the bacteria is growing, turn it, and then onto the next video.